So the 19th chapter in the first PUC is the excretory products and their elimination. So what is excretion? Excretion is the elimination of nitrogenous waste from the living tissues of an organism called excretion. That is the definition. It should come from the living tissues of an organism, that is called the excretion. And the organisms excrete urea, ammonia, uric acid, and even the Na plus, K plus, Cl minus also will be excreted. And the organisms which excrete their nitrogenous waste products in the form of ammonia called ammonotelic organisms, and the phenomenon called ammonotelism. Example, I will give you code word PCMB A square. Protozoans, crustaceans, coelentrates, C, molluscans, bony fishes are the ammonotelic organisms. The organisms, and this ammonia, it requires large amount of water for its excretion, and it is toxic substance. And the urea is also toxic substance. The organisms which excrete their nitrogenous waste products in the form of urea called urotelic organisms, and phenomenon called ureotelism. All mammals excrete urea, and even the terrestrial frogs, terrestrial amphibians, toads, they excrete urea, and even the cartilaginous marine fishes, they excrete urea. The organisms, and this urea also requires large amount of water for its excretion, and it is toxic substance. And the organisms which excrete their nitrogenous waste products in the form of uric acid called uricotelic organisms, and the phenomenon called uricotelism. I will give you code word AIR. Airs, insects and reptiles are uricotelic, along with the land snails are also uricotelic organisms. And then there are certain structures, excretory structures. Say for example, malphizian tubules are present in the cockroach, are the excretory structures. Not only that one, and also uricoses, nephrocytes are also the excretory structures of cockroach. This is very important. And then the nephridia, segmental, integumental and pharyngeal nephridia are the excretory structures of the earthworm. And the flame cells called protonephridia, they are the excretory structures in the platyhelminthes. So protonephridia or flame cells can also be seen in, so far not asked this question, so that is in rotifers, few annelids and cephalochordata members, that is amphioxus. That is also very important for your NEET examination. And green glands in prawn. Green glands are also called antennal glands. They are the excretory structures in case of prawn. That is also so far not asked. Those questions not asked, very important for the NEET examination. And NEET examination is very easy, provided if you read complete textbook, as I am repeatedly telling, that the textbook of biology is only the book, and don't refer any other books, only for the NEET examination in biology. Textbook. Textbook is the Bhagavad Gita for the NEET examination. So the excretory organs in case of human beings are the kidneys and the skin. Kidneys are located in the abdominal cavity, one on either side of vertebral column. Exactly after this last rib, the last rib where it reaches there, the kidney located. Actually, the last rib it just touches the kidneys. So kidneys are located in the abdominal cavity, one or either side of the vertebral column, reddish browny in color, bean shaped structures are the kidneys, and about 10 to 12 centimeters in length and 2 to 3 centimeters in thickness, and 120 to 170 grams in weight. That is also very important. 120 to 170 grams in weight, 2 to 3 centimeters in thickness, and 10 to 12 centimeters in length. That is also very important as far as the kidneys are concerned. And from the inner margins, ureters arise and they open into urinary bladder, which in turn open into urethra. Urethra is common for both men and women. Renal arteries supply blood to the kidneys, whereas the renal veins collect the deoxygenated blood from the kidneys. And then this is about the kidney structure. And longitudinal section of the kidney shows outer cortex and inner medulla. So in the medulla region, there are about 15 to 20 pyramid-like structures called renal pyramids, and the basal portion called calyx or calyces. Calyces open into pelvis, pelvis open into ureter. One more important thing is the region of cortex which extends into medulla in between renal pyramids called columns of Berrettini, important for your NEET examination. That is the columns of Berrettini. And kidney composed of millions of small structures called nephron. The nephron is the unit of excretory system. And this nephron consists of cup-like structure called Bowman's capsule and proximal convoluted tubule, henal slope, descending limb of henal slope, ascending limb of henal slope, distal convoluted tubule, and collecting tubule. A tuft of blood capillaries present in front of Bowman's capsule called glomerulus. Afferent glomerular arteriole brings blood to the glomerulus, and the afferent takes away blood from the glomerulus. That is also very important. And there are two types of nephrons. There are cortical nephrons and juxtamedullary nephrons. In case of cortical nephrons, Bowman's capsule placed in the cortex region. A little portion of a henal slope can be seen there, and even the vasa recta may be present or absent. Small amount of, the region of vasa recta can be seen or even absent, that is in cortical nephrons. And the which runs henal slope, which is small in case of cortical nephrons, henal slope long and runs throughout the medulla. They are called juxtamedullary rays. And the afferent glomerular arteriole, which takes away blood actually from the glomerulus, and then it spreads over the proximal convoluted tubule and distal convoluted tubule to form the capillaries over the walls of the PCT and DCT. Later it forms a renal vein, but peritubular capillaries of vasa recta can also be seen. The blood capillaries around the henal slope called the vasa recta, they form the peritubular capillaries. And coming to the structure of the glomerulus, and the formation of urea takes place in the liver, and from the liver it comes to the heart, then along with the post-caval vein, and from heart to lungs after the elimination of carbon dioxide. So 18 liters of carbon dioxide eliminated per day. That is very, very important, 18 liters of carbon dioxide. So then this urea, it along with the blood comes to the left auricle and then left ventricle, to all parts of the body urea goes, whereas 25% will reach the kidney. A question may be asked like this, the percentage, the least percentage of urea present in this, or the highest percentage of urea present in pulmonary artery, like this, that question may be asked. That is also very important. And this urea thus formed from the liver and reaches the kidneys. In the kidneys, formation of urine takes place. This involves 3 steps: glomerulus filtration, tubular reabsorption, tubular secretion. Glomerulus filtration, as it takes place under pressure, it is called ultrafiltration. Ultrafiltration. So afferent glomerular artery or afferent arteriole brings blood to the glomerulus. The wall of the glomerulus, here also very important, it is composed of squamous epithelium, and that is line called endothelium. And then it is in contact with, it comes in contact with the epithelial cells, that is the podocyte cells of the Bowman's capsule. In between the endothelium and the epithelial cells, that is the podocyte cells of Bowman's capsule, there will be basement membrane, and the filtration takes place across this, the endothelium, basement membrane, and the epithelial cells of podocyte cells of the Bowman's capsule. So these podocyte cells are arranged in an intricate manner so as to leave some spores called slit spores. This is also very important. So podocyte cells in the Bowman's capsule, they are arranged in intricate manner so as to leave some pores called slit pores. Very important. And then as the filtration takes place under pressure, it is called ultrafiltration, and it is the GFR, glomerulus filtration rate. And about 1100 to 1200 ml of blood is filtered during the glomerulus filtration per minute. So this amounts to 1/5 of the blood pumped from the ventricle of the heart per minute. That is also very important, so far not asked. So 125 ml of blood will be filtered per minute, or 180 liters per day blood will be filtered during the glomerulus filtration. And this, the filter rate, it enters into the capsular space of Bowman's capsule. This capsular space of Bowman's, in the capsular space of Bowman's capsule, filter rate contains the salts, amino acids, glucose, urea, water. Unfiltered RBC, WBC, and large sized proteins like albumin enter into efferent glomerular arteriole, which capillarizes over the walls of PCT and DCT. Later it forms the renal vein, and along the vasa recta, also peritubular capillaries. Later it forms the renal vein. And this is about the filtration. And at the junction of this DCT and afferent arteriole, a sensitive part called JGA region is formed. Juxtaglomerular apparatus. It is a sensitive region formed by the cellular modification of the distal convoluted tubule and afferent arteriole. This is also very important. A cellular modified region of the distal convoluted tubule and the afferent arteriole. At this junction, it is formed a sensitive region called JGA. And this filtrate, as it enters here, and then this filtrate contains some useful substances like amino acids, glucose, and even the sodium are reabsorbed into the blood capillaries which surrounds the PCT and DCT. And here the PCT and DCT, about 70 to 80% of tubular reabsorption takes place, and the glucose and amino acids are reabsorbed along with the sodium by active transport, and some nitrogenous waste are absorbed by the passive transport. And this is known as tubular reabsorption. As it takes place at the expense of energy, say for example amino acids, glucose and sodium, it is called active transport. 70 to 80% of this kind of tubular reabsorption takes place in the PCT and remaining in the DCT. Both in PCT and DCT, tubular reabsorption takes place. In both these cases, the hydrogen, ammonia, potassium are added. That is both in the PCT and DCT, and also maintains pH. That is also very important. As the renal fluid flows to this primary urine, remaining salts, urea, as it flows to the descending limb of Henle slope and Henle slope. This Henle slope, it allows the permits water entry but do not allow to enter the electrolytes. And then as the renal fluids of two different concentrations flow in an opposite direction, this creates a counter current multiplier system. And as I was telling you, this in the cortical region, the concentration of the filtrate, that will be about 300 osmomolar liter inverse. And then as it comes to the PCT, it is about 600, 900 and 1200 at the Henle slope. Salt concentration will be more at the Henle's loop, and then the ascending limb of Henle slope, again it decreases from 1200, 900, 600, 300, like this it will be enters into the DCT. In the DCT, as I told you earlier, the tubular secretion takes place. Hydrogen, ammonia, potassium are added. Creatinine in certain wastes are added. And in the collecting tubule, absorption of water takes place. And because of these three steps, glomerulus filtration, tubular reabsorption, tubular secretion, the product formed is called urine. So this urine, 95% of it consists of urea and 2.5% of salts and 2.5% of salts and 2.5% 95% water and 2.5% urea and 2.5% other salts are present in the urine. So while passing through the PCT and DCT, water is also removed by osmosis, as the concentration of water decreases when amino acids and glucose are removed. Hence, the water is also removed from the region of higher concentration, lower concentration by osmosis. There is one hormone called ADH, antidiuretic hormone. It is also called vasopressin, secreted in the posterior lobe of the pituitary gland, that is the neurohypophysis. Regulates the water composition. It stimulates the uriniferous tubules PCT, DCT for the absorption of water. The undersecretion of ADH causes the diuresis, that is the loss of excess of water in urine, that also takes place. And if the body fluids exceeds certain osmoreceptors will be there, and these receptors reduce the secretion, decrease the secretion of ADH by feedback mechanism, feedback mechanism. And this ADH is a vasoconstrictor, and this increases the GFR, glomerulus filtration rate, GFR. There are other things which regulate the working mechanism of the kidneys called RAS system, that is the renin, aldosterone and ADH antidiuretic hormone, and this system is called RAS system. And the renin, as I told you earlier, JGA is the sensitive region formed by the cellular modification of the DCT and afferent arteriole at the junction of this DCT and the afferent arteriole, a sensitive region formed because of cellular modification called JGA. JGA secretes renin, the hormone. So this renin spelling, mind the spelling, RENNIN is the enzyme secreted by the gastric juice, but here it is the hormone secreted by the kidney, RENIN. This is also very important. This RENIN, it helps in the conversion of angiotensinogen to angiotensin 1 and 2. Angiotensin 1 and 2 are vasoconstrictors, especially angiotensin 2 is a vasoconstrictor, and it increases the glomerulus filtration rate. Thus, aldosterone also help in the removal of sodium hormone. That hormone helps in the removal of sodium in the distal parts of the renal tubule, that is distal convoluted tubule. Thus, renin and then aldosterone, angiotensin. So these things, they play an important role in increasing the glomerulus filtration rate, that is called RAS system. And one more thing is for how the NaCl is removed. And how the NaCl will be removed means, as the renal fluid flows through the ascending limb, from the ascending limb of the Henne's lobe, then it goes to the descending limb of vasa recta. From the descending limb, ascending limb of vasa recta, and ascending limb of vasa recta to interstitial region, that is NaCl. Whereas the urea will be transported from the ascending limb of Henne's lobe, and to interstitial region from the collecting tubule, that is the urea. So far not as these things are very important for your examination. And coming to the last aspects, as I told you earlier, this urine is slightly acidic in nature, and the pH is 6, and 6 is the pH acidic in nature, and 20 to 25 grams of urea is excreted per day, as I told you earlier, and the 18 liters of carbon dioxide eliminated per day. And similarly, sweat glands excrete sweat. Sweat consists of urea and NaCl. And sebaceous glands produce sebum, which consists of the steroids, waxes, hydrocarbons are present in the sebum. So thus the excretion takes place. And there are certain abnormalities caused because of the elevated levels of the urea called uremia. Uremia can be corrected by dialysis. One method called hemodialysis. Hemodialysis is a process in which the blood of a patient is removed and allowed to pass through the dialyzer, which removes the urea and adds some useful substances. While the blood is taken out, heparin is added, anticoagulant, and the same blood is allowed to enter into the blood after into the body after dialysis. Then antiheparin is added. This is the very important method of dialysis, that is for to remove the urea followed in the world, that is called hemodialysis. In the world, almost all people, they suggest this kind of dialysis for the removal of urea. And inflammation of the glomerulus is called glomerulonephritis. And sometimes the accumulation of calcium oxalate salts called kidney stones, called renal calculi. And this renal calculi is highly painful. That is called, pain is called colic pain, and renal calculi is formed by calcium oxalate. So any seeded fruit, say for example, doctors will tell the seeded fruits like the guava and then tomato especially contain calcium oxalate. They block the ureters, and these such stones are called kidney stones, that is called renal calculi. So this is about in detail the chapter 19 excretory products and their elimination. Please go through each and every sentence. Please subscribe.